Hello, good morning, everyone. This is Leah with Headwaters Science Institute, bringing you another Lunch with a Scientist presentation today. And before we get into meeting the wonderful Abby Miller from UNR, I just wanted to give you a very special announcement. So as you know, we've been talking about our student research experience, which is starting on February 2nd, coming up very soon. And just to get the rest of you to sneak in for that last registration opportunity, we've actually extended our deadline to this Friday, January 29th. If you haven't already seen it, our executive director, Meg, made a special announcement about this on our social media this morning. But the research experience is a great opportunity for high schoolers to pair with a professional scientist mentor, create a science project of their very own, author a research paper, and create a research presentation. It's a perfect thing to add to a college application, and we know that it's college application season right now for our high school juniors. So jump on the registration while you can. The program will likely fill up. You can find it on our website. So to move on to our presentation today, I am really excited to introduce Abigail Miller. She's a first-year graduate student in the Voyles lab at the University of Nevada, Reno. And her research centers on wildlife infectious disease, particularly in regard to frogs and amphibians. She graduated from the University of California, Santa Barbara in 2018 with a degree in zoology, and she later spent time working as a research assistant studying host-pathogen interactions at the Salk Institute in San Diego. So today, she's going to talk to us a little bit about how emerging wildlife diseases are becoming more prevalent and looking at treatment and host pathogen interactions in wildlife and amphibians around the world. So I'm very pleased to welcome Abby Miller.
Hello.
Hi, good morning. Thanks for being here.
Thank you for having me.
You're very welcome. So we're really excited to hear more about amphibians and infectious disease and how all that works. But before we get into that, you and I were talking before the broadcast about how you are a UNR student. And there's a little bit of research that's still possible in your lab, even with the pandemic going on. I know that that's a little bit tricky as a research scientist. So can you tell us how that's working right now?
Yeah, so I think that the main difference is that it kind of just requires a lot more scheduling to make sure that there's not like in one area. So it's definitely different, and I think it also requires a lot of compromise and like being aware that things are going to move a little bit slower, but that's okay as long as everyone's safe and things like that.
Mm-hmm. And I would imagine it's pretty critical that you continue your research so that you can continue studying, right?
Yeah, exactly. Especially, yeah, I'm a PhD student, and so that always takes quite a bit of time. So, yeah, we can't delay for too long. Especially when you already kind of have ongoing projects in the lab. It's hard to kind of just stop altogether.
Mhm.
So yeah, it's been interesting.
I bet. Well, we're really, really excited to hear about the research that you've given us a little teaser of. So I will let you jump into your presentation. You can go ahead and go full screen.
Cool.
Very excited to see you talk.
Thank you. Yeah, hi, my name is Abby Miller. I am a first year PhD student in the Voyles lab at UNR, and we study wildlife disease, especially in regard to frogs and amphibians. And so today I'm going to talk about disease ecology, starting with some main concepts, and then going into a little bit of detail about my study species, and ending with some past and my current research. So to start, disease ecology is the field of biology that deals with disease mechanisms and dynamics in different populations. And so these are just some pictures that represent some current wildlife diseases. These pictures are showing Tasmanian devils, little brown bats, and golden frogs. And so disease ecology can include things like mapping disease spread following host pathogen interactions and disease persistence in wild populations over time. Disease ecology is really important because wildlife disease can limit biodiversity and decrease genetic diversity over time. And so to start off with some main concepts of disease ecology, we have the disease triad. The disease triad refers to the environment, the pathogen, and the host. The pathogen is any infectious agent or a harmful microbe, something like a bacteria, a virus, or a parasite. The host is going to be the individual that's affected by the pathogen, and the host can be symptomatic to infection, meaning that they can display symptoms like coughing or sneezing, or asymptomatic to infection, where they are infected by the pathogen, but they don't actually display any symptoms or sickness behaviors. And then lastly, you have the environment, and the environment is going to be the physical area that's actually bringing together the host and the pathogen, and allowing for disease transmission and ultimately infection. And one of the main aspects of infection in regard to the host is the immune system. And so the immune system generally has two main branches. It has the innate branch and the adaptive branch. The innate immune system is considered any aspect of the immune system that is non-pathogen specific. And so this can be something like your external skin barrier, which is going to stop pathogens in general from entering the body. But it's not targeting any one specific pathogen in general. Or mucous membranes, which have a similar effect. And you also have innate immune cells. And these are going to be cells within your body that are working to attack and kill pathogens in the body, but are triggered by general patterns of infection. And are non-specific. So they're also not targeting any one specific type of pathogen. And so, for example, an innate immune cell can recognize when a pathogen makes it past your external barriers and enters the body, but that innate cell does not recognize exactly what type of pathogen it is. So it's going to attempt to attack and kill the pathogen, but it doesn't get any external information to help. And then lastly, you have the microbiome, which plays a large role in innate immunity, and I'll go into that in a little more detail in a minute. And then you have the adaptive immune system. So the innate immune system is a lot faster to react than the adaptive immune system, but the adaptive immune system is typically more effective, and that's because it is specific. And so this means that these B and T cells that make up the adaptive immune system can be triggered by a specific type of pathogen that matches up with specific receptors and other mechanisms that these immune cells have on their cell surfaces. And that can allow for much faster clearance of the pathogens. And so, like I said, the microbiome is involved in innate immunity. So our immune system helps to fight against pathogenic microbes, but our body actually also houses millions of symbiotic or helpful microbes. And the microbiome refers to these good or helpful microbes that help the body. And the microbiome is essential for maintaining homeostasis or just optimal function. And the microbiome can help in functions like digestion. And so the microbiome is also considered an innate immune defense because it can help the body defend against pathogens. And it can do this by taking up space in the body and outcompeting pathogens, or by actually releasing chemicals and things like antibiotics that can actually harm and kill pathogens. So if your microbiome was not functional, you could have a higher disease susceptibility and a lesser immune function overall. And so moving forward, I'm going to go into more of a broader look at wildlife disease and my study species in particular. And so the prevalence of wildlife disease is actually currently increasing through time, and that should be concerning for many reasons. The first of which is zoonotic diseases. And zoonotic diseases are diseases that are spread from animals to humans. An example of this that we should all know is COVID-19. And human activity can actually increase zoonotic diseases. And this can happen through activities that increase human contact at wildlife borders that previously had little to no human contact, which can allow for novel spread of zoonotic diseases. And climate change can also affect disease. And so this can happen by changing climates, forcing animals to move from their current habitats to find new suitable habitats. And this can lead to more disease spread, again, in places that diseases were previously not found. So another reason that we should care about wildlife disease is because it can cause biodiversity loss. And biodiversity loss can affect trophic interactions. And so a trophic chain is something like a food chain. And this figure here is showing an example of the structure of a trophic chain, where at the bottom you have plants and primary producers, and then above that you have herbivores and things that eat plants, and above that you have things that eat herbivores, and so on and so forth. And so if a member of a trophic chain is removed, this can have cascading effects on the rest of the entire ecosystem, especially in regard to those levels that were above and below that level that was removed. And so now I'm going to focus a little bit more on amphibians in particular here, because this is what I study. And amphibians are particularly important members of their communities. And this is in part because they can be considered bioindicator species. And this has to do because of their very sensitive skin. And so amphibian skin is very sensitive because it is permeable, and that's because it is involved in functions like maintaining water and ion balance, as well as respiration in many cases. And so amphibians can be considered early warning signs of an unhealthy or polluted environment because of their sensitivity of their skin. Also, many amphibians have two life stages, and in many cases, they have both an aquatic and a terrestrial stage. And so they can be important for nutrient transfer between these two aquatic and terrestrial habitats. And so amphibians are currently experiencing a decline that is larger than that of any other group of animals, and that is in part due to wildlife disease. And so I study a wildlife disease called chytridiomycosis. And chytridiomycosis is a disease that is caused by a fungal pathogen. The fungal pathogen is called Batrachochytrium dendrobatidis, or abbreviated BD. And BD is a fungus that will affect that sensitive skin of amphibians and can eventually lead to cardiac arrest. And so this figure here is showing the life cycle of the fungus BD. There are two life stages. There is a zoospore stage, which is a mobile stage, and a sporangia stage, which is a immobile stage. And so the sporangia immobile stage will create more zoospores, which will be released from the sporangia and go to infect new hosts. And so the spread of BD has thought to have been amplified by human travel and other human activities. And there's also differential risks in different environments. And so in environments, for example, that have higher elevation and cooler temperatures, there's typically thought to be a higher risk. And so now I'm going to transition into a little about my past and current research plans. And so I had my first research experience when I was an undergrad in college, and I joined a lab that was working on a project focused on finding new treatments for BD infection. And we looked at two different species of frogs. And so we worked with that blue species, which is Dendrobates tinctorius, and then the green species, which is Pseudacris regilla. And so the central question of the project was focused on finding better treatments for BD infection. And so the most widely used treatment for BD infection is called itraconazole. And itraconazole is an antifungal, and it's highly effective at treating BD infection, but it needs to be used for at least 5 days in a row. And it can be toxic to frogs, so it needs to be used in very dilute doses. And so because of this, the lab that I joined was interested in testing a new antifungal treatment called BMP. And this new antifungal was chosen because it was non-toxic to in trials with healthy frogs, and it would only need to be used one single time. And so we started with in vitro tests of BD clearance. And so in vitro refers to an experiment that's done not in a living animal, so in something like in a petri dish. And so these graphs are showing the clearance of BD zoospores in this blue graph and sporangia, the immobile stage, in the purple graph. And we used varying dilutions of BMP, that new antifungal, so that is shown on the x-axis. And you can see that, especially in regard to this blue zoospore graph, even fairly dilute doses of BMP, the new antifungal had a clearance effect on BD zoospores. And so because of that, we moved on to an in vitro trial and an in vivo trial. I don't know if I just said that. In vivo refers to an experiment that is done in a living animal, for example, in a frog. And so these graphs are showing 3 different colored lines. And these 3 different colored lines refer to different treatment groups for these frogs. So these black lines are referring to the control treatment group. And so that means that these animals were infected with BD and then untreated. The blue treatment group is referring to the BMP treatment group. So animals treated with the BMP, the new antifungal. And the yellow lines refer to the animals treated with itraconazole, the older antifungal. And again, we use these 2 different species of frogs, and the y-axis is showing pathogen load. And so pathogen load means the relative amount of BD that is present on the frogs. So a higher pathogen load on this y-axis means that they have more BD on the frog. And so overall, especially if you're looking at the Dendrobates graph, this blue frog, you see that the BMP blue lines, the slope of those lines don't look very different compared to the black control group. And so this shows that the BMP treatment actually was not effective at clearing BD over time. And you can compare those black and blue lines to that yellow itraconazole line, where you see that the pathogen load was decreasing over time as a result of that itraconazole treatment. And so this was interesting because it contrasted with our results from the in vitro trials. And as well, because you can see a slight difference in how these 2 different species were reacting to these different treatments. And that suggests there might be some interspecific differences of the treatment and the BD infection, which could relate to different immune function in these different species. So overall, our new antifungal was not effective at clearing BD in vivo. However, new and more effective antifungals are still necessary to develop. As we gain more knowledge on the amphibian immune system, we can better develop treatments for BD infection. And so lastly, I'm going to discuss my plans for my PhD project, and as well as give some supplementary data from previous experiments as they relate to my project plan. So like I said before, the microbiome is an important aspect of host innate immunity, and knowledge on the immune system will help us create better treatments for BD infection. And so because of this, I'm interested in generating germ-free frogs, which are frogs that have no microbiome, in order to see how they respond to BD infection and how exposure to the microbiome during frog development can contribute to immune system development, with the goal of better understanding frog immunity and BD infection over time. And so in frogs specifically, the microbiome has many functions, including protection from pathogens, assisting in metamorphosis, metabolism, and digestion. And so, for example, during metamorphosis, the frog is transitioning from a tadpole diet to an adult frog diet. And the microbiome can actually change in composition during metamorphosis in order to better aid in digestion of that adult frog diet. And so we know that the microbiome does all these things in frogs, but we don't quite know its full effect on frog immunity. So how can we test the importance of the microbiome? Well, we can remove the microbiome to see how the immune system can function in its absence. And there are no previous experiments that have generated germ-free frogs. However, there are a few experiments that have reduced or depleted the microbiome, and looked at disease susceptibility as a representation of immune function. And so this is an example from a paper that did that, where researchers depleted the microbiome of tadpoles by using antibiotics. And then they allowed those tadpoles to develop and go through metamorphosis and later become frogs. And then they infected those adult frogs to look at disease susceptibility and immune development. And these are some results from that experiment. And so these first two graphs are showing the microbiome depletion of those tadpoles. And the first graph is showing the gut microbiome, and the second is showing the skin microbiome. And you can see in both of those graphs, there are those three treatment groups that have had their microbiome depleted, and that is shown by a reduced bacterial diversity on that y-axis. And then this last graph is referring to the infection with the adult frogs. And so again, those three treatment groups that had their microbiome depleted had a higher establishment of that pathogen that they were infected with present in their gut. And so these findings suggest that this early life disruption of the microbiome can have effects on the immune response over time. And so, to summarize, frogs are keystone species in ecosystems, and they are currently at risk due to infectious disease, which is in part due to human activities. The microbiome is critical for frog immunity and susceptibility to disease, but the actual extent of this is currently unknown. And so my PhD project is focused on further deciphering the effects of the microbiome on susceptibility to disease and immune system development, with the ultimate goal of applying these concepts to generate better treatments for BD infection, as well as used for management of wild populations. And I'd like to thank the Voyles Lab and UNR EECB, the Briggs Lab that I did my undergrad research in, and the Headwaters Institute for this opportunity. Thank you.
Thank you very much, Abby. That was great.
Thanks.
So we're going to open it up to some audience questions here in a minute. And I want to jump off with just a couple questions that came up for me while you were speaking.
Yeah.
The first of which was, so we talked a lot about the impacts of these fungal infections in frogs. And I wonder have scientists seen the presence of fungus on these frogs increase over time due to climate change or some other human impacts? Or have frogs always been susceptible to these kinds of infections?
Yeah, that's a really good question. So the history of the BD fungus is actually pretty interesting. It's correlated with these frogs that were actually used in human pregnancy tests back in the past. And so these frogs were transported all around the world, basically, and they think that that's correlated with the spread of the fungus all around the world. In regard to like climate change and things like that, we're not particularly sure how exactly that's going to affect the spread of the disease and transmission. But yeah, the fungus typically grows better in cooler temperatures. So that's something to keep in mind. But how that's actually going to look in practice, I'm not quite sure yet. So we'll have to see.
And so we think that it popped up after this pregnancy test experiment happened?
Yeah, it was thought to have started in these frogs. No, it's not like 100% sure that is what happened, but that is one hypothesis. And yeah, they can actually test museum specimens and things like that to see if there is any prevalence of the fungus on them, and kind of track the emergence, like the year, the decade, and where the spread popped up in different places.
And so now in developing these new antifungal treatments, I know that it's very common in science that we try things and they just unfortunately don't work. And we have to redo our experiments or our hypotheses and we move forward. So typically, going back to that experiment that you did, what would be the next step after that first antifungal treatment didn't work? Maybe developing a new one, or how would you go about that?
Yeah, that's a great question. I think that yeah, there was BD clearance in those in vitro tests, so I think that it's not a lost cause. So you can definitely go back and see if there's a more effective way to administer the antifungal. Maybe the way that we put it on the frogs wasn't as effective, um, or you can see that the BMP treatment that we used is actually a solvent and good for increasing the efficiency of other drugs. So you can see if adding that BMP treatment in conjunction with another antifungal could increase effectiveness. Yeah, there's lots of different things that you can try to do. It is frustrating, but yeah, that's science. It's not always going to work out perfectly.
Hmm. And then moving forward, when you do find a treatment that works, because you're dealing with wildlife, how do you administer a treatment on a large scale when you might not know where to even find all these frogs or you have to go search for them?
Yeah, that is one of the issues with itraconazole, the older antifungal that people generally use, because you have to use it at least for 5 days in a row, and so you can bring it out to the field and try to administer it to as many frogs as you can find, but you have to keep going back and doing that for 5 days in a row, and so there's no real promise that you're getting those same frogs for those 5 days in a row. And so, yeah, it's just you got to do your best and do what's available to you right now. But yeah, it's definitely complicated when you're working with wild populations, I think.
Hmm. So I'm wondering, you talked a lot about why biodiversity is important, and of course, we're trying to prevent the decline of amphibians in particular because it's so rapid. I wonder why you chose in particular to study amphibians.
Yeah, it was a little random, I think, to start. I was an undergrad in college, like I said, and I just got an email to the listserv that they were looking for research assistants. So that was kind of my like jump into the world of amphibian disease research. But I just think frogs are really awesome. They're so diverse in like all of their behaviors, especially in like reproductive behaviors and things like that. So as I continued to take classes, I got more and more interested in it. Yeah, I feel like what's not to like about them? They're great.
Hmm. So, a question coming in here from the audience, going back to the treatment that I just asked you about, are there implications for other species or the habitat when treating wild populations of frogs?
Yeah, that's definitely something to keep in mind. Because frogs, a lot of the time are aquatic, you have to test how much of that antifungal treatment is going to be dispersed back into the environment and if that's going to have effects on other species, especially if you're working with an antifungal like itraconazole that can be harmful in larger doses. And so that is something that we were looking at with that new BMP treatment. I wasn't involved with that project in particular, but I know generally they applied that BMP treatment on frogs and then tried to rinse them off with water and then test the water and see how much of the treatment actually came off. To try to answer that question. So yeah, that's another thing that I think makes dealing with wild populations very challenging, because you have a lot of sensitive species that depend on each other in very intricate ways. And it is very difficult to try to address that when you're working in the lab. But it's important. It's very important to think about.
Hmm. So we've got a great follow-up question here. We talked about why you chose amphibians to study, but taking it deeper, why did you become interested in infectious disease specifically?
Yeah, I feel like that's a hard question too. Yeah, so like I said with the undergrad, that just happened to be the project that I was working on. And then again, as I have been taking more and more classes, I think I'm pretty interested in the immune system, just because I think it is very complicated and infectious diseases can differ so much and cause these huge cascading effects, and they can affect like animal behavior and reproduction and all this other stuff. And so I think that that is kind of overwhelming, but that's kind of why I like it too. I think that it's such like a complicated problem to address, and pathogens are so diverse too. I think I might have already said that, but yeah, that's kind of my was my main interest.
Mm-hmm. And then going to the other end of the spectrum, if there's, say, a high school student who knows right now that they really want to study infectious disease or conservation biology or zoology, what path would you recommend that they take to become a PhD student like you are?
Yeah. So I was a zoology major in undergrad, but I think that like any major within biology, I think that you can set yourself on that track. I think that in undergrad, you have a lot of opportunities to kind of email professors and just try different labs and see who has a spot for an undergrad research helper. I volunteered in a few different labs. And it's okay to try something and not like it too. There's no harm in just emailing your professors if they talk about their research and you're interested in that. I think the worst thing that someone can say is, no, I don't have space for someone. So yeah, that's what I would recommend. I think that yeah, your undergrad time in college is like the perfect opportunity to just try as many things as you can possibly think might be even a little bit interesting. And and yeah, I mean, I'd be happy to help with a more specific answer to that question too, if someone wanted to email me about that. But yeah, that's kind of what I did.
Mm-hmm. That's great. So I think we're going to wrap up in a minute here, but I wanted to end on a fun question. What's your favorite frog that you have come across?
Oh man. I really like those, the blue frogs that I had in the presentation, those Dendrobates frogs, and that is because I really like frog reproductive and parental care behaviors. I think that it's super interesting. And those blue frogs, they actually they have their tadpoles, and then the male will give them like a piggyback ride, and it'll go and like hide their tadpoles in little pools of water like all around the forest. Um, and they have incredible memory. They have to remember where they put every single one of their tadpoles. And I just think it's cute that they give them a piggyback ride too.
Mm. So then one more follow-up to that. Once travel is allowed again, is there some future potential for you to go and see these frogs on location?
I would love to. That would be great. Yeah. My lab does a little bit of research in Panama, so that could be cool. But yeah, we'll have to see. I'm okay with anything. So playing them by ear.
Great. Well, big hopes for the future. Thank you so much, PhD student Abby Miller from UNR. Thank you for talking to us about infectious disease and amphibians. We really appreciate it.
Yeah, thank you so much for having me.
Have a great day.
Thanks.